I guess we're live on with everyone. I'm Tim Block, president and co-founder of the Hepatitis B Foundation as Blumberg Institute. Conrad, can you hear me, Mala?
Yes, I can hear you.
Oh, good. And Juto. All right, so look, I just want to say a few words. I didn't realize in the waiting room would be my video, so that was a little embarrassing, but just want to say a few words before I turn it over to Dr. Jutao Guo, who's our Executive Vice President for Research, and appropriately enough for today's talk, the London Professor of Experimental Therapeutics here, an endowed chair set up by the London family, Tom London and Linda London. Well, first of all, thank you all for joining us today. This is an experiment for us. When we had here at the Blumberg Institute, created by the Hepatitis B Foundation, let me just close the door, created by the Blumberg Hepatitis B Foundation, we've had a great seminar series here. We're a nonprofit research institute looking for hepatitis B and liver cancer and Hep D drugs, and we've had a great seminar series here. So when we had to suspend that because of the COVID pandemic, that was extremely hurtful to us, and we wanted to continue it. We decided to put it online just a couple months ago. Normally, the speakers visit here, we get to meet them, and that's a great experience. And we are going to have to take that up again and get our speakers here to visit us personally, individually. But in the meantime, we're going online. And when we decided to go online, Jutao Guo suggested we put it online and make it available to the entire Hep B community. The International Hepatitis B meeting was also suspended this year, and there's definitely pent-up appetite for our seminars dealing with Hep B, liver cancer, and Hep D. So I thought that was a great idea. We all thought that was a great idea of Jutao's to put this online and make it available to the whole HBV community. So now I understand we have more than 300 people who've registered. We just put the word out, figuring this out this week. That is quite a multiple of our usual audience, Mala. So we're really pleased that our first speaker of this series is Dr. Mala Maini, someone we've been wanting to get here, still not here, but I was glad that she immediately agreed to give the talk. So I will say this, please, audience, check online at the Hep B or Blumberg website if you want to continue on. We have a series set up for June. We'll continue on with speakers every month, and maybe we'll keep our seminars online so they're always available. Next week is Dr. Scott Friedman from Mount Sinai, who's going to talk about liver fibrosis and liver fibrosis markers. And then we're going to have Marion Peters, who will give the annual Bruce Whitty lecture. She's from UCSF. And she'll be talking about precision medicine for Hep B. So with that, I will stop and turn it over, or I'll try to turn it over. Jutao, thank you. I guess, do I leave now? I gotta leave my place. All right. Oh, wait. I want to say one more thing, Jutao. Don't mute me yet. I have this book here. So Dr. Maini is turning over her honorarium. We do pay an honorarium to our speakers back to the Hepatitis B Foundation, which is very generous. But we got to give her something. So we will be sending Dr. Maini the book that Dr. Blumberg wrote called Hepatitis B: Hunt for a Killer Virus, and it's a great story. I doubt you have it because we have the 20 last copies available, and we give them out very stingily. And so we'll be sending this to you with a couple of other tokens of our appreciation. Thank you very much, Malay. I got to leave this. I couldn't log on. That's why I was, of course, just learned how to use our television a couple weeks ago, and I couldn't get online. So I'm going back to my desk where I hope it's going. Thank you, Jutao. Now mute me.
Okay, thank you. So welcome everyone to the first Hepatitis B Foundation and the Blumberg Institute's web-based seminar. So in this series of the webinars, we were inviting the leading investigators in hepatitis B research and also related liver disease research to share their cutting-edge discoveries as well as their thinking and exploration to cure chronic hepatitis B. So the recent cure of the hepatitis C with direct-acting antivirals really inspired people more seriously think about cure of the chronic hepatitis B. However, I actually strongly believe that, for achieve a functional cure of chronic hepatitis B, we must to activate or restoration of the functional innate adaptive immune response against hepatitis B virus to maintain a constant surveillance of the residual infection. So we are very pleased that Dr. Maini accepted our invitation and kick off our first seminar. So Mala is a professor of viral immunology and the deputy head of departments in University College London. Is also is a consultant physician in viral hepatitis clinic. Mala received her PhD in T-cell immunology in 1998 at University of London and established her research laboratory in 2002 to work on HBV immunopathogenesis. Over the years, she has achieved a lot. As a recognition of her excellent achievements, she was promoted to a personal chair in 2009, awarded a Wellcome Trust Senior Investigator Award in 2013, and elected fellowship of the Academy of Medical Sciences in 2016. So Mala's lab was at the forefront internationally in research of HBV immunopathogenesis. And her published very extensively in last 2 decades and cover many aspects of the HBV immunology through dissect the functional defects of the T cells, HBV specific T cells, B cells, as well as NK cells. And I was very impressed by her studies, especially on the metabolic control of the T cell function, and also her recent discovery about functional defects of the HBV specific B cells. So, and she also, tried to harness all these fundamental discoveries for develop the therapeutic vaccine or use harness those mechanisms for develop the therapeutics to nurture the host immune response towards the treatment of the chronic hepatitis B. So without further ado, let's just welcome Dr. Maini to present her seminar. Thank you.
Thank you very much. So thank you so much for this invitation to Tim, Jugao, and everyone at the Bloomberg and the Hepatitis B Foundation. I'm really honoured to be kicking off your new virtual seminar series. I hope somebody's going to let me know if I cut out. And could I now have my slides put up, please? So what I plan to do once the slides are up is to break off the talk at a couple of points during the presentation to allow you to ask questions, because I think it will be difficult for me to keep going talking to a blank screen for 45 minutes, and probably difficult for people who are listening in to keep listening. So I think you can ask questions either by posting them into the system or putting up your hand and speaking to me directly during the presentation or at the end of the presentation. So can you see my slides? Jugao, can you just, so the slides are advancing by themselves. Can you set it up so that I can advance them myself? Okay, let's see if that works now. So I think with this audience, I don't really need to tell you what a massive problem hepatitis B is. I'm sure you all know that it still is persistently infecting more than 250 million people worldwide, causing an estimated 2,000 or more deaths a day from the complications that it triggers of cirrhosis and hepatocellular carcinoma. And actually, Tugao, can you keep the presenting the picture down at the bottom of the slide, if that's okay, the bottom right, because I've set up the slides generally to keep that space clear. Thanks. So the two chief barriers to cure in hepatitis B are the viral features, which are the persistence of the CCC DNA, the episomal form of the virion, and the fact that it frequently integrates into the chromosome. And both these forms are resistant to all current antiviral drugs. On top of that, we have a profound level of immune exhaustion due to the persistent high viral load, which chronically stimulates the immune system for decades, and the fact that the virus infection takes place within this unique tolerogenic liver niche. So alongside all sorts of exciting developments for new antiviral drugs, why would we need to think about an immunotherapeutic approach? Well, I think that most of us would agree that even with the best new antivirals that are being developed, and even with combinations of these, we're very unlikely to be able to eliminate every last trace of HBV in the liver, particularly the cccDNA and integrated DNA that I just mentioned. But we do know that most adults who are infected with this infection can maintain any little residual traces of the virus under very successful long-term immune control, such that we only see reactivation of HBV in these naturally resolved donors if they're given very profound immune suppression. So they provide a blueprint for the potential role of immunotherapy in helping to maintain residual virus under good control. And we do know also that even once chronicity has been established, HBV does remain susceptible to immune control. And we've seen that from natural experiments where people have had to receive bone marrow transplants from resolved donors. So we can think about the goals of immunotherapeutic approaches in terms of short and long-term goals. In the short term, we could use immunotherapeutic approaches to act in tandem with antivirals to actually help to speed up the clearance of infected hepatocytes. And this could be done with either non-cytolytic or cytolytic removal of infected hepatocytes. But in the long term, I think the role of the immune system is particularly important to provide robust immune surveillance to limit any viral reactivation and spread from any residual cccDNA. And also to provide immunosurveillance to prevent carcinogenesis from the integrated DNA. So I've made an analogy to try to describe the different ways that we can think about boosting the immune response. And I think that the immune response can you see my pointer, by the way? Yes, no? I can't.
No, there's no point. There's no pointer right now.
Okay, so if you think about the immune response in a patient with chronic hepatitis B being rather like that very battered Fiat Cinquecento that you see in the upright picture, it's really on its last legs. And the question is, can we do something to revive this old car? Could we put our foot down on the gas pedal and get it going again by revving it up? Because there's a lot of data suggesting that in hepatitis B, some of the cell-intrinsic immune responses that we rely on for initial control of viruses are not being triggered adequately. So perhaps just by triggering those innate or cell-intrinsic immune responses, we might be able to get the immune system going again by putting our foot down on the gas. Another approach to recovering the inherent endogenous immune response, the old battered Fiat Cinquecento, is by trying to take the brake off and trying to release responses that have been too tightly controlled. And I'll talk more about these approaches. But if these fail, what we can do is think about bringing in a completely new model. So this nice shiny new Ferrari could be used to replace the body's own response to hepatitis B and bring in new responses, either by triggering those by, say, a therapeutic vaccination, which gets the body to make new responses, or by adoptively transferring either cells or antibody infusions. So just quickly to talk about replacing the endogenous adaptive immune response with a new CAR, because this is not something we predominantly work on, but we have over the years collaborated a lot with Antonio Bertoletti, who's leading the work along with Ulla Protzer's lab in using T-cell receptor redirected T-cells as a form of exogenous therapy. So here the T-cells have a new T-cell receptor inserted, which is one that's able to recognize HBV peptides presented by the MHC complex of infected cells. And these can then be infused into the patient as a form of adoptive cell therapy. So these papers describe how this work was first developed and then used in a first-in-man study. Lion TCR, Antonio Bertoletti's company, is progressing this exciting approach in the setting of HBV-related hepatocellular carcinoma. And in this paper, we've described the knockdown of PD-1 on these TCR redirected T cells as a way of allowing them to survive longer in the tumor liver environment. CAR T cells, of course, are another way of approaching this adoptive cell therapy. And then there's another approach which we've been collaborating on, which looks very interesting, with the company Immunocore, which uses a bispecific reagent, which is a soluble T cell receptor, which is affinity matured, again, to be able to recognize particular HLA-restricted HBV peptides, fused to an anti-CD3 molecule, which then allows it to harness the local T cells regardless of their T cell receptor specificity. And then on the other side of the equation, we can also think about replacing immunity with infusions of humoral immunity. So therapeutic antibodies are being developed for testing in hepatitis B, and those can either work by neutralizing the virus or harnessing cells for antibody-dependent cell cytotoxicity, or they can be made to be bispecific to deliberately trigger cells such as T cells in the environment. But mostly what my lab has been focusing on is seeing whether we can find ways of reviving and releasing the endogenous adaptive immune response, so the patient's own immune response. So trying to see what we can do with this battered CAR to rescue it. And this diagram just summarizes some of the key approaches that are being used and examined in the field to try and do this. And I think that having a good, very immunogenic therapeutic vaccine is a backbone of most of these approaches, because it helps to then focus any immunotherapeutic approaches on the right antigens and make them as specific as possible. So just to briefly talk through these, we can think about reducing the antigen load in order for immune therapies to become more effective, or we can think about ways to actually specifically tailor the immune response by checkpoint inhibitors, metabolic rescue, and blocking inhibitory cell types. And most of this work has been looked at in the context of T cells, which I'll come on to shortly. But I want to first start by focusing in on B cells and look at whether it's possible to rescue the endogenous B cell response. So what do we know about the importance of humoral immunity in hepatitis B? For a long time, antibodies were just assumed to be important in protecting against reinfection, but it's becoming clear that humoral immunity actually plays a very important role in the ongoing control of chronic viral infections. And this has been nicely exemplified by a lot of work recently in HIV, where development of therapeutic antibodies has been shown to have a very potent effect. And it's also clearly exemplified in the case of hepatitis B by the effect of rituximab, which is being given for various malignancies and rheumatological disorders. So this is a B cell depleting infusion, which can result in either a flare-up of existing chronic hepatitis B or reactivation of resolved hepatitis B. So that tells us that some aspect of B cells or of the antibodies that they produce are critical for maintaining control of hepatitis B. And we can think about the potential roles of the antibodies as neutralizing or by harnessing other cells through ADCC, ADCP, or a vaccinal effect on dendritic cells. But remember that B cells don't just produce antibodies. They don't just develop into plasma cells and pump out antibodies. They're also very important for antigen presentation and for producing cytokines. So a few years ago, we set out to try and ask what was going on with antigen-specific B cells, particularly B cells targeting the surface antigen in hepatitis B, because we know that by definition in chronic hepatitis B, we can't detect anti-HBs antibody or minimal levels of it. But we wanted to know, even though we can't detect the antibody, are the cells that can produce the antibody, those memory cells, still present in patients? And if they are they then going to be amenable to some kind of therapeutic rescue? So this work was led by Alice Burton, who at the time was doing her PhD in the lab, and was done in collaboration with Nadège Pelletier at Roche, who developed a bait for us, which was a FITC labeled HBs antigen, which allowed us to then stain just those B cells that have the B cell receptor on their surface that's binding to hepatitis B surface antigen. And then because they were then fluorescently labeled, we could look at them directly ex vivo and phenotype them. So for the first time, we could actually study straight out of the patient what the surface antigen-specific B cells looked like. And we made the surprising finding that in a large proportion of patients with chronic hepatitis B, even though we couldn't detect anti-HBs antibodies, we could detect the memory B cells that are capable of producing the antibody. And in fact, we found those S antigen-specific B cells at similar frequencies in patients, no matter whether they had chronic hepatitis B, resolved, or had been vaccinated. So this suggested that the defect was not at the level of the persistence of these cells, but at their function. And so what we did was here you can see in this FATS plot, this little population in the red box is the cells that have actually stained with the bait. So these are the B cells that have the right B cell receptor to bind to the surface antigen. We sorted on the flow cytometer just those specific B cells, cultured them in vitro to turn them into plasma cells, and then looked in the supernatant to see whether they were able to produce anti-HBs antibodies. And when Alice did this experiment using healthy donors who'd been vaccinated, their S antigen binding B cells were all very good at producing the antibody, as you would expect. But instead, the chronic patients' S antigen-specific B cells were completely defective. So although they were present and staining the bait, they were unable to produce the antibody. So because we could then stain these cells directly ex vivo, we could start to look at them and see what defects they might have that might be limiting their function. And this is just some initial profiling that Alice did to look at their memory phenotypes. And the large majority of these S antigen-specific B cells in vaccinated donors fell into this upper right quadrant, which is the phenotype of conventional memory cells, 2721 positive. But instead, when we looked at the chronic hepatitis B donors, you can see this expansion of red here. In these are multiple different donors arranged by age. So there's not actually an effective age, but there's a large expansion of atypical memory B cells in red here. And these, if you can see in this red quadrant on the FACS plot, have downregulated both CD27 and CD21, a component of the B cell receptor signaling complex. So what are these atypical memory B cells? Well, they've been described in a number of settings where there's a very high dose chronic antigenic stimulation, and they've got a lot of features in common with exhausted T cells. They have, as I mentioned, downregulation of CD21, which is an important component, signaling component of the B cell receptor, but they have upregulation of a lot of inhibitory molecules. And you can see that in our patient samples here in the red. that these atypical memory B cells had upregulated a number of important inhibitory molecules, and amongst these, you can see at the end here, PD-1 was highly upregulated. So we already knew that PD-1 was highly expressed on T cells in hepatitis B, but this was the first demonstration that it was also increased on the B cells. In addition, when we looked further at the phenotype of these cells, we saw that they had aberrant expression of homing molecules. So they had reduced expression of CXCR5, which would be important for getting them into the lymph node to get the adequate help from T follicular helper cells. But instead, they had upregulation of CD11C, which tends to push B cells towards inflammatory sites. And so we wondered if they might be homing to the liver instead of lymph nodes. And in fact, we were able to look at this from our access to HBV-infected liver tissue. You can see in the right-hand histograms that whereas in control livers, we were unable to stain any essential-specific B cells, in almost every hepatitis B-infected liver that we looked at, we could see these antigen-specific B cells accumulating locally in this organ, which is obviously, I think, the first time that antigen-specific B cells have been shown to accumulate in a non-lymphoid tissue in humans, and is not what you would normally expect. Now, some very nice work came out from Antonio Bertoletti's lab at the same time. So they had coincidentally been carrying out a sort of parallel study looking at a lot of the same stuff as us. That's what comes from working together for too many years. They made strikingly similar findings regarding the dysfunction of S antigen-specific B cells. But what they then went on to do, which is very nice, shown here, was to compare the core antigen-specific B cells with the S antigen-specific. And we know that, of course, a core antigen-specific response, it doesn't seem to be dysfunctional in hepatitis B. So anyone who's exposed to the virus makes a good anti-core antibody response. And in line with that, you can see that on the left here that in paired stains, in every case, there's more core-specific B cells than S antigen-specific B cells in the same donor. And then when you look at their phenotype, the atypical B cells are really only expanded in the S antigen-specific B cells. The core-specific B cells in green look very similar to the S antigen ones in vaccinees. So they've got low levels of atypical B cells. And again, when you look at antibody production, the core-specific B cells are good at producing antibodies, just like the S antigen-specific B cells from vaccinees. So this seems to be predominantly a defect of the S antigen-specific B cell compartment. What can we do to rescue those cells? So both Antonio's group and ours tried targeting PD-1 because that was an obvious target, having seen that it was upregulated on these cells. And we did this in combination with stimulation through CD40 ligand and IL-21. And you can see here that we could get an increase in the cytokine production from these B cells, a decrease in their propensity to undergo apoptosis, marked by the expression of annexin. And then from Antonio's lab, you can see that they also increased their capacity to produce anti-HBs antibody with these culture conditions. So just to summarize this bit of the B cell work, we showed that persistent hepatic tropic viral infections like hepatitis B drive an accumulation of these atypical or dysfunctional S antigen-specific B cells in both the blood and the liver, which then have impaired antiviral capacity. I haven't shown all the data, but we showed they have impaired signaling, homing, survival. They are poorly able to differentiate into plasma cells, reduced production of antiviral cytokines, and crucially of the anti-S antibody. And this is all in contrast, but it starts to provide some initial molecular targets that could be exploited for boosting more effective humoral immunity for the functional cure of hepatitis B. Okay, so before I move on to now talk about T cells, is anyone out there wanting to ask me a question about this first part of the work?
Yeah, it seems no one raised a hand.
Okay, I'm just looking at all the messages. There's lots of people having problems with the sound. Sorry to hear about that. Is there anything we can do about that?
Or someone lose the sound?
Oh, there's loads of messages saying there's issues with the sound, but hopefully most people are okay. I'll carry on then.
Yeah, recently that's internet, because a lot of people is using is heavy loaded, is very unstable.
Okay, I'll carry on. So, okay, so I'm now going to move on to talk about hepatitis B specific T cells, particularly CD8 T cells, and what can be done to try to rescue the endogenous response. So the most obvious place to start is looking at checkpoint inhibitors like PD-1, where you're taking off the brake on the T cells. And this is obvious to start with because it's been used so successfully in cancer immunotherapy. And the other side of this equation is to actually put the foot down on the gas pedal more by using co-stimulators, which can be molecules like we've studied, like IL-12. And I'm not really going to go through this part of the work. I'm just going to very quickly mention the justification for these checkpoint inhibitors. So multiple studies have been done, principally from Carlo Ferrari's lab, but we've also characterized some of these different checkpoint inhibitors that are highly upregulated on HBV-specific T cells, particularly the ones in the liver. And we know that a lot of the ligands are highly expressed by other cell types in the liver. On top of that, there's some nice fundamental work in animal models showing that if you combine therapeutic vaccination with checkpoint blockade, you can enhance the response to the vaccine. That was done initially with LCMV, and then from studies from Michael Roggendorf's lab with the woodchuck model of woodchuck hepatitis virus. And the next step's been taken with this small study done in hepatitis B infection using the PD-1 blocker nivolumab with or without therapeutic vaccination, which it was only a single dose, single low dose, but it showed some promising early effects in the sense that one patient seroconverted, and I think out of 30, most of them had a small drop in S antigen levels. But I want to talk about going a bit beyond just these surface checkpoints like PD-1, CTLA-4, TIM-3, and thinking about trying to revive the T cell in a more fundamental way by replenishing other aspects that it needs. So in particular, thinking about filling up the gas tank with more fuel. But obviously, when your car's in trouble, there's all sorts of things you can do to try to rescue it rather than just ramming a foot down on the gas or taking off the brake. So what do I mean by this? So, for example, there's a lot of work emerging from both a study that Anna Zurich did in my group, but also from several other groups in mouse models and tumor settings, showing that exhausted T cells are characterized by quite fundamental defects in their mitochondria. So they tend to have highly dysfunctional mitochondria, which limit their flexibility in terms of metabolism of glucose. So they're unable to switch between glycolysis and oxidative phosphorylation in order to maximize their bioenergetics. And this is really fundamental to mounting a strong, effective, and rapid T cell response, where you need T cells to undergo multiple rounds of proliferative expansion and to produce lots of effector molecules. So they're having efficient bioenergetics and underlying metabolism is really key to that. So I'm just going to talk about a recent study that Leah Swadling in the group did, which has uncovered a role for Topher G in maintaining this mitochondrial fitness. And we did this in collaboration with Katia Simon, who's an expert on autophagy in Oxford. And so autophagy, as you all know, is a constitutive, dynamic, lysosome-mediated intracellular bulk degradation process. And it has two main roles. On the one hand, it's important for maintaining cellular homeostasis. So particularly mitophagy, where you can recycle dysfunctional and damaged mitochondria to produce repaired new mitochondria is very important. But by breaking down these intracellular particles, it can also be very important for biogenesis, for example, by lipophagy. And we can now start to look at this process by flow cytometry on the small populations of cells that we're studying in the human immune system, because there's a reagent available that an antibody that binds to LC32. And LC32 is a lipidated form of this cytoplasmic protein, which once lipidated is then incorporated into new autophagosomes as they form and as they become encapsulated. And so by staining the levels of LC32 and then blocking degradation of the autophagosome using baflamycin, we can get an idea about autophagic flux in individual cells. Just to summarize very briefly what Leo found, when he just stained for the levels of this autophagic protein LC32 directly ex vivo using cells that he isolated from the blood and the liver, he noticed straight away that there was much higher levels of LC32 expression on the cells in the liver compared to the blood. And then breaking this down further, if you look in these FACS plots, the cells that are highlighted in red are the ones that we know have a phenotype of tissue residence. That means that they're exclusively found in the liver because they express both CD69 and CD103. And those cells had particularly high levels of LC32 expression, suggesting that tissue residence was associated with upregulation of autophagy. We can look at this with another method. So by image stream here, you can see in the upper panel again that there's really nice LC32 fluorescence seen in combination with cells that are expressing CD69 and CD103, whereas the CD8 cells in the lower panel that don't have CD103 and have low levels of 69 also have lower levels of LC32. And so here, using this technique, Leo was able to not just look at the intensity of expression, but also count the number of puncta of these autophagosomes. And so taken together, these data show us that the tissue-resident population of T cells is highly adapted to have increased levels of autophagy. And what might be the use of this? So we looked to see whether the autophagy high T cells were able to better maintain mitochondrial fitness, as you would expect, given the role of autophagy and mitophagy in doing that. To do this, Leo stained with two mitochondrial dyes, mitochondrial red and green tracker here. And the cells that are in the red box are the ones that have depolarized mitochondria. So you see a lot of those in dead cells, and you don't want to have too many of those in your live cell gate. But what we saw was that generally in T cells that are just going in and out of the liver, so just the liver infiltrating T cells, were very prone to have depolarized mitochondria, perhaps because of the increased oxidative stress that you see in the liver environment. Whereas the T cells that were resident in the liver had very low levels of depolarized mitochondria, suggesting that they were protected from mitochondrial damage by their autophagy. And to just confirm that, Leo used a drug which is able to inhibit autophagy by blocking the nucleation of the autophagosome. And he could then see that when you use that, you get a striking increase in depolarization. So in terms of therapeutic application of these findings, we looked to see whether there were any cytokines that could reproduce this liver effect that could be used perhaps therapeutically. And out of all the different cytokines that we tested, Leo found that IL-15 was potently able to induce autophagy in a dose-dependent manner, which was then blocked using anti-IL-15 blocking antibodies. And this is interesting because it's known as a prototypic liver cytokine. And in fact, we were able to isolate primary hepatic stellate cells from liver explants and show that these cells could also support T cell autophagy And again, this was in an IL-15 dependent manner, because when our anti-IL-15 was added, you lost the effect of the stellate cells. So just to summarize this part of the talk, we've compared the cells that are coming in and out of the liver transitorily with those that are long-term resident, and found that the liver-infiltrating cells are very prone to have depolarized mitochondria, which is associated with the impairment of their bioenergetic functionality. But on the other hand, the tissue-resident cells have adapted to their environment by increasing their LC32 expression and their basal autophagy, allowing them to protect the mitochondria from depolarization. And this can happen in an IL-15 dependent manner. And IL-15 I haven't shown you, but it can also drive the tissue-resident expression of these key markers to maintain T cells in the liver. Again, I'm happy to stop at this point if there's any questions about the autophagy coming through. I can now see quite a few questions about B cells, which I maybe come back to at the end now. Does anyone want to interrupt with a question about the metabolism data?
Seems we can continue.
I'll carry on. Okay.
Accumulate question to the end.
Yeah, that's fine. Okay. All right. So finally, just to think about, because up till now I've been talking about cell intrinsic mechanisms of regulation. But what about other cell types that might be present in the liver that could be constraining the functionality of the T cells? And we've worked on this respect on both NK cells and myeloid-derived suppressor cells. And here with the analogy, you can think about, instead of the Ferrari coming in to replace the Fiat Cinquecento, it's actually bashing it up and taking it off the road. And this is an actual headline that I picked out of an Italian newspaper. So this is trying to think about whether we can remove destructive immune cell players that might be limiting the functionality of T cells. And we've, for a number of years, been interested in the role of NK cells in this respect. So NK cells can, of course, help T cell responses by producing cytokines like interferon gamma, which actually can boost the T cell response. But an increasing amount of data suggests that they can also really constrain T cells. And this seems to be particularly relevant in the liver. The NK cells are very prevalent in the liver. Up to 30% of the lymphocytes in the liver are NK cells. And they upregulate some death ligands like TRAIL in the setting of hepatitis B infection, which they don't normally express in the healthy liver in humans. They're going to be brought into incredibly close contact with the infiltrating T cells, because they've got to make their way through this sinusoidal network, which you can see hopefully in this plaster cast of the liver, with these very narrow lumen vessels permeating in a fine meshwork through the liver, which will push the resident NK cells and T cells into intimate contact. And natural killer cells, of course, are well known for their killer capacity. You can see some nice images we took of these with a colleague who's an expert in scanning EM. And what our previous work showed was that not only do the NK cells, particularly the liver resident ones, upregulate death ligands like TRAIL, but the hepatitis B specific T cells particularly upregulate receptors for this ligand, so the TRAILR2 receptor. And so that makes them highly susceptible to deletion by NK cells. And so what we're doing now with Mariana Diniz, a senior postdoc in the group, in collaboration with Ellie Barnes's group in Oxford, is to test whether we can enhance the response to a very nice HBV vaccine that Ellie's just published on, and it's in fact using the same ChAdOx backbone as the Oxford vaccine that's being developed for SARS-CoV-2. We're collaborating together to see whether in an in vivo model, whether we deplete NK cells or block their regulatory function, can we enhance the immunogenicity of the vaccine by increasing the T cells that respond to that vaccine. And then finally, I just want to wrap up by stressing the importance of these tissue-resident memory T cells that I've mentioned in passing. This was work that was published by Laura Pallett from the group a few years ago, been identified in many other tissues in humans and mice, and increasingly shown to have a critical role in frontline immune surveillance. These are cells that don't recirculate. They stay poised at the site of infection locally in the tissue, and they're very good at mediating rapid effector function. They can often be long-lived, and I've shown you that they have this increased autophagy and mitochondrial fitness that might contribute to that longevity. And we're able to study them because of our great collaborations that we've set up over many years with a number of hepatologists and surgeons to allow us to get access to fresh liver tissue. And I want to say a big thank you to all the people here and many others at all these different sites who work so hard to help us to collect all this liver tissue and to analyze it fresh directly ex vivo, and also to mention incredible hard work of my team to process these samples that come in at very uncivilized hours. So here, just to recap, this is the identification of the tissue-resident CD8 T cells that Laura made, where on the bottom flow panel, you see in the top right-hand quadrant, it's quite difficult without a pointer, you see cells that are co-expressing CD69 and CD103, which you don't see in the blood at all. So these are seen in all different types of liver samples, no matter how we obtain the tissue, healthy margins, perfusates of liver transplants, but we don't see them coming out into the blood. And not only do they have this unique surface phenotype, they also have a particular transcriptional phenotype with high levels of BLIMP. They have unique phenotypic and functional features which indicate their adaptation to the environment. Very high levels of PD-1, which should constrain their functionality. And yet when you stimulate them, even just for 4 hours directly ex vivo, they pump out cytokines, and important cytokines like interferon gamma, which of course has anti-HBV activity, and IL-2, which we think will be really critical for overcoming liver tolerance and allowing them to be long-lived. So importantly, in the context of HBV, these tissue-resident T cells are still expanded in the liver. In fact, they increased further in HBV livers compared to healthy control livers, and they still don't come out in the blood. So we can't monitor these tissue-resident T cells by taking blood samples. And what we see is that if we look at their frequencies according to the outcome of HBV infection, those people who have good control of their HBV with very low levels of viremia are the ones who have the highest levels of tissue-resident T cells, suggesting that they are indeed playing a role in immune control. How are they retained in the liver? Well, they have a number of molecules that should contribute to this. The expression of CD69 is a negative regulator of S1PR1-mediated T cell egress, and CD103 is an integrin which allows them to bind to ecadherin. And in this immunostaining, you can see that we found that hepatocytes in the human liver heavily express ecadherin. So through their contact with hepatocytes, either directly or through the fenestra of the liver sinusoidal endothelial network, T cells should be able to be tightly adherent to that. And to test how adherent they are, we looked to see whether they were egressing at very low frequency into the hepatic vein. So this is work done in collaboration with Xavi Fawns in Barcelona and with Dominic Yu at the Royal Free Hospital, where we were able to obtain blood samples from the hepatic and portal vein. I'm just showing actually the hepatic vein here. And unlike liver samples like the liver perfusate, where you can see the tissue-resident T cells down at the bottom, the hepatic vein, with or without balloon occlusion, looks exactly like the peripheral blood. So there's no low-level egress of these tissue-resident T cells detectable into the hepatic vein. And then just recently, this work's just in press, we've taken advantage of a very interesting situation in humans where we often have liver transplant being taken place with MHC mismatch of donor and recipient. Because the liver is such a tolerogenic organ, there's no need for MHC matching. And that allows us to track donor and recipient lymphocytes and leukocytes and see how long they persist in the liver. And so Laura and Alice have done this work along with the whole team, where we've looked at allografts that have been removed after a certain number of years and found that we can still find the donor leukocytes, both donor tissue-resident T cells and donor Kupffer cells, persisting for more than 10 years in the human liver. And in addition, we could see that some recipient T cells and mononuclear phagocytes could infiltrate the liver and start to acquire some of these characteristics of tissue residence. So this, just to wrap up, really stresses the importance of monitoring the liver as well as the blood in ongoing trials of functional cure in hepatitis B. We've got compartmentalization of HBV-specific T cells, liver-resident T cells. We've also got liver-resident NK cells and many other cells that are really enriched in the liver. And of course, we've got the viral reservoirs, compartmentalized cccDNA and integrated DNA. And we've done some work with Uppy Gill and Patrick Kennedy to show that in the setting where biopsies are becoming harder and harder to access, we can get some very nice immune monitoring with fine needle aspirates instead of biopsies. And these fine needle aspirates, which are done with a very narrow bore needle, it's a lumbar puncture needle, are pretty painless. They can be well tolerated for repeated sampling, and they do actually allow us to sample the important cell types. So here you can see in these examples that in blood, the liver aspirate from the fine needle and the biopsy, we can see the tissue-resident T cells are present in the aspirate and the biopsy, but not in the blood. And the HBV-specific T cells are much more frequent in the fine needle aspirate and the biopsy than the blood. And here's another example where we've got HBV-specific T cells directed against these different pools of HBV peptides with really nice responses present in the fine needle aspirate. I think I'll just skip the last slide because I want to leave lots of time for questions, but this is just showing that we can also sample live hepatocytes in parallel with leukocytes on these fine needle aspirates. So just to summarize now, I've shown you that immunotherapy of chronic hepatitis B can aim to either revive or restore or replace the failed endogenous response. Defective S antigen-specific B cells do persist in many patients with chronic hepatitis B, so they may be amenable to therapeutic rescue. Dissecting the intrinsic and extrinsic constraints on T cells has identified multiple targets beyond just the classical checkpoints. And liver-resident T cells are retained long-term and highly adapted to the niche, so they're important to consider for immune monitoring and therapeutic targeting. And I'm going to wrap up by thanking my amazing team who worked so hard to produce all the data I've shown. I've listed the names here and I've talked about most of them who've gone along, and also all our amazing collaborators and funders. And now let me start to tackle some of your questions. So how do you want to do this?
So before we go to these questions from the chat, I think that let's break these questions into the sections. So we start from the B cell dysfunction.
Okay.
So before that, I first ask a more general question. So in B cell response, it's quite normal actually for the core antigen, right? For polymers or other viral protein. So that was dysfunction is only seems primarily for the surface antigen. Of course, that surface antigen is a secreted protein, but core antigen, that was the antigen load actually also is very high, right? But and from the cell-based study, a lot of the negative core, negative capsid, they may can also released through a rather different way to the circulation. So then why just the surface antigen specific antibody was dysfunctioned, but not others?
Well, I would imagine it's a dose effect because the amount of circulating S antigen is vast. It's in excess of any other known protein in humans, I think. So we're talking microgram quantities of the S antigen. And as far as I know, the core antigen's not circulating at anything like that level. So I would think the first postulate most likely would just be the level of triggering of the receptor on those cells is far outweighed by any other. But there may well be other effects, because we did see some defects in the global B cell population as well. So there's likely to be other differences to do with, I think, the amount of help from T cells as well is critical. So the S antigen specific response is very dependent on T cell help, and we know that both CD4s and CD8s, and so that would include the T follicular helper response, is defective in chronic hepatitis B. So that lack of help probably plays more of a role for the S antigen specific antibody response.
So Anna may have some question about on the B cell dysfunction. So his question is: so are those B cells similar to those in patients who had acute infections? That is, genetically, would they produce the same IgG subtype against the surface antigen if they could? So what's that really mean? I think that, for example, if the B cell tolerance, that was, we know that the B cells may have produced high affinity antibody, lower affinity antibody. It's only a subpopulation is exhausted and someone even deleted or something, if that's related to this affinity.
Yeah, I mean, these are tricky questions. I mean, in acute infection, I mean, we only looked sort of in a couple of donors, and we haven't really been able to look very early in acute infection to see whether the S antigen specific B cells start off being highly functional and then become dysfunctional. I would postulate that that would be the case, but it's always very difficult to. The acute patients that we study are always the ones who then go on and resolve. It's really tricky clinically and immunologically to study acute going on to chronic, which is what would be the really interesting comparison to make. And in terms of the details of looking at the specific B cells directed against other antigens and a fine epitope mapping of those responses, that's all work that remains to be done. It's really at the beginning of this investigation.
So I see there's Tim's question.
Yeah, Tim's question is a very interesting one. I mean, because I guess you're saying, well, if the B cells are so defective, well, why would it matter if you, in a chronic patient, if you then take them away with rituximab, why should you get a flare? And you're right, that suggests that although they are defective and we can't detect much antibody, they obviously are doing something. So it could be that they are producing some antibody and it's being soaked up by the S antigen, and it's in a very tight sort of equilibrium, so that when you take them away, you tip it in the wrong direction. But probably they're also playing other roles that I mentioned at the beginning in terms of maybe antigen presentation, cytokine production. So the fact that you get those flares in chronic patients point towards other effects, I would say, as well.
So next one is that Robert Close. Do we think antibodies in HBV have important effector functions in eliminating infected cells via NK cells and other effectors?
I mean, there's a little bit of work done on that from some mouse model studies where people have modified the FC receptor region, used therapeutic antibodies, modified the FC receptor region, and shown FC-dependent effects, suggesting NK cells and phagocytes and other cells may be very important. So it's not just about neutralization. And that certainly seems to be the case in HIV. There's a lot of studies confirming that you need this cellular harnessing role of antibodies, not just neutralizing roles. But really in humans with hepatitis B, we don't know that much yet. We're actually just looking into what cell types are present in the human liver, which could, and what capacity they have to bind to antibodies to participate in those type of roles at the moment.
So next one is Brett's titry. So I think that I translate the question and also incorporate my own questions into it. So generally speaking, unlike viral drug treatments, you just suppress viral application. And this immunotherapy usually is a double-edged sword, especially for the HBV infection, because the immune response can eliminate, control virus infection, and also can cause liver damage, right? So what's how we can fine-tune this, response? We just take the advantage and avoid that harmful tissue damage.
Yeah, I mean, that's $1 million question. It's always the worry with immunotherapy. I mean, especially since we know so little about what proportion of hepatocytes are infected and are actually presenting the key epitopes in different patients. In some there may be lower levels of either infection and or antigen presentation, such that you would only, even if you had a very strong directed T cell response, you'd only eliminate a proportion of the hepatocytes, and then you'd be able to get repair. But in others, there may be very widespread antigen presentation, and any kind of directed T cell response would need to be titrated very carefully. There is also the possibility, of course, of targeting non-cytolytic T cell mechanisms. So relying on cytokines, which can help to eliminate hepatitis B without killing the target hepatocytes. And that's a way of sort of amplifying the response without doing too much damage. But this is just it is a very tricky balancing act. So far, different immunotherapies that have been tried, including PD-1 blockade, haven't caused overwhelming liver damage, which is reassuring. But every new approach needs to be very carefully evaluated from that point of view.
So Chris Moore is raised his hands. So Chris, can you just speak out your question? Do you find the audio button? Hi, Chris.
Hello.
I can just about hear something.
Can you hear me? Can you hear me?
Yeah, I can hear you. Yeah. Hi, Chris. Hi.
Hey, Mala. How are you?
I'm fine. How are you?
I'm good. I hope you're faring this pandemic okay over there.
Yeah.
I have a question. So, let's put aside the debate about whether a checkpoint inhibitor or that checkpoints are involved in HBV tolerance for a second. So, let's assume that they are, and let's assume that a checkpoint inhibitor would work in some patients for HBV in terms of breaking tolerance and potentially leading to some curative responses. If you had a molecule that only did that in the liver, that only blocked the tolerance mechanisms that you are very clearly showing in a lot of your work in the liver, but did not have effect in the blood, so the molecule was staying only in the liver. If you broke tolerance in the liver, would that be the same as a systemic breakage of tolerance for HBV? Would it be as effective in your opinion?
Well, I mean, you could argue it would be more effective, potentially. I mean, certainly it should be safer in some respects because you would avoid some of the systemic toxicity of risk of autoimmunity in other organs. But we do know that obviously molecules like PD-1 are important on the global T cells in the liver, not just on the virus specific. So you wouldn't get round that issue of potentially unleashing a very strong local T cell response.
But based on what you've learned in the liver, is there anything mechanistic in the liver that needs to be specifically targeted in terms of breaking tolerance that we can't necessarily do if in the plasma or in the blood?
Well, I mentioned a couple of things which I think are not totally specific to the liver, but are more enriched in the liver. Things like some of the regulatory populations, which are really enriched, their interactions with the NK cells and myeloid-derived suppressor cells. We've previously looked at IL-10, which is enriched in the liver. So I would say, I mean, most of these things actually, you can detect them in studies in the periphery, but they're just really exaggerated in the liver.
Okay. Thank you.
So we still have one more question that from Dr. Tianlong Zhu. So can B cells recognize HBV peptide present on the hepatocyte surface?
No, they don't recognize peptides. They recognize a whole antigen through their B cell receptors. So when a peptide's been presented in the context of MHC, then it will be recognized instead by a T cell. Is that what you meant?
Yeah, I believe that's, of course, antibody can, certain antibody can recognize that, right? But,
I mean, it can recognize the whole, if the whole antigen is presented on the surface as it's budding out of the hepatocyte, maybe that's what you meant, then of course the antibody can bind that. And it's debatable how much that occurs in hepatitis B. There's certainly some staining showing surface antigen bound to the surface of hepatocytes. And even if it's in the close vicinity of the hepatocyte, if the antibody then binds, you could get antibody-dependent effects by that way.
Okay, thank you very much, Mala. That's a very impressive seminar. So, you will have, chats with our faculty.
Okay, great.
Thank you very much for everyone to attending this webinar, and also welcome you join us in our next webinar. Thank you very much.
Thank you. Thank you for the invitation.
Yeah, thank you.
I think you have